Welcome back to G Fun Facts Online. Today, we are diving deep into a discovery that fundamentally rewrites the rules of how the human body heals, or specifically, why it fails to heal. We are talking about the heart, the organ that beats roughly 100,000 times a day, every day, until the moment we die. It is a masterpiece of biological engineering, but it has a fatal flaw. Unlike your skin, which can knit itself back together after a deep cut, or your liver, which can regenerate from a fraction of its original mass, the adult human heart is strictly non-regenerative. When a heart attack strikes, the tissue does not regrow. It dies. And in its place, the body builds a tombstone. That tombstone is a scar. It is a stiff, non-contractile patch of fibrosis that weakens the heart's pumping ability and paves the way for heart failure, a condition that eventually claims the lives of nearly half of those diagnosed. For 50 years, the central dogma of cardiology has been that this sequence is inevitable. Once the heart muscle dies, it is gone forever. But a groundbreaking discovery published in late 2025 has shattered that assumption. Researchers have identified a specific RNA molecule, dubbed TY1, that does not act as a building block, but as a commander. It triggers a process known as transcriptional healing. This mechanism bypasses the need for stem cells entirely. Instead of adding new cells, TY1 rewires the immune system's response to injury, turning off the alarm that causes scarring and turning on a hidden pathway for repair. This is the story of that molecule, the decades of failure that led to its discovery, and the revolution it promises not just for heart attacks, but for the entire field of medicine. To understand the magnitude of transcriptional healing, we must first understand the enemy, which is the scar itself. When a coronary artery is blocked, oxygen starvation or ischemia sets in. Cardiomyocytes, your heart muscle cells, are voracious consumers of energy. Without oxygen, their mitochondrial power plants fail within minutes. As these cells die, their membranes rupture, spilling their internal contents into the surrounding tissue. To the immune system, this spill is indistinguishable from a bacterial invasion or a viral attack. The body's defense forces, specifically macrophages and neutrophils, rush to the scene. Their job is twofold. First, clear the debris. Second, seal the breach. In their frenzy to contain the damage, they unleash a cytokine storm of inflammation. They digest dead cells, but in the process, they also damage bystander cells. Crucially, the heart cannot wait for slow regeneration. It is a high-pressure pump that must beat 60 to 100 times a minute. A weak spot in the wall could burst, leading to immediate death, so the body prioritizes speed over quality. Fibroblasts are recruited to lay down collagen, a tough, fibrous protein. This is the scar. It is a biological patch, strong enough to prevent the heart from rupturing, but stiff and dead. It cannot contract. It cannot conduct electricity. As the remaining healthy muscle tries to compensate for this dead weight, the heart enlarges and eventually fails. This is the mechanical reality of heart disease. For decades, science has thrown its most advanced tools at this problem. We have tried plumbing, using stents and bypasses to restore blood flow. We have tried electricity, using pacemakers to maintain rhythm. We have even tried the holy grail of regenerative medicine, stem cells. This brings us to the second part of our story, the stem cell mirage. The road to the discovery of TY1 is paved with the disappointment of the stem cell era. In the early 2000s, the scientific community was electrified by the idea that we could simply inject fresh stem cells into a damaged heart, where they would take root, differentiate into new muscle, and restore function. It was a beautiful, logical theory. And it failed. In trial after trial, the injected stem cells vanished. They did not integrate. They did not become heart muscle. Most were flushed out by blood flow or killed by the hostile, inflammatory environment of the infarct. And yet, a curious phenomenon persisted. Despite the disappearance of the cells, Some animals, and later, some human patients, showed a modest improvement in heart function. If the cells were not staying, how were they helping? The answer came from the laboratory of Dr. Eduardo Marban, a pioneer in cardiac research. His team realized that the stem cells were not bricklayers building a new wall. They were generals shouting orders. Before they died or washed away, they secreted tiny, chemical-filled sacs called exosomes. These sacs contained a soup of proteins and RNA molecules, a paracrine signal that told the native heart tissue to survive and repair. This realization birthed the concept of cell-free therapy. If we could bottle the signal, we would not need the cells. But exosomes are messy. They contain thousands of different molecules, varying from batch to batch. To make a consistent drug, researchers needed to find the one molecule in that soup that mattered. They spent years sifting through the molecular noise. Finally, deep within the exosomes secreted by cardiosphere-derived cells, which are a potent type of heart progenitor, they found it. It was not a protein. It was not a standard messenger, RNA. It was a small, non-coding RNA molecule from a family known as yRNAs. They synthesized a refined version of it and named it TY1. The mechanism of TY1 is what separates it from every therapy that came before. It does not try to force cells to divide. Instead, it targets a fundamental, ancient immune trigger that goes wrong during a heart attack. That trigger is the cGAS-STING pathway. Let's break this down, because this is the core science of the discovery. Deep inside every cell, DNA is supposed to stay in two places: the nucleus and the mitochondria. When heart cells die during a heart attack, their nuclear envelopes dissolve and mitochondria burst. DNA leaks into the cytoplasm, the cell's liquid body. The immune system has an ancient sensor for DNA in the cytoplasm called cGAS, or cyclic GMPAMP synthase. Evolutionarily, this sensor exists to detect viruses, which often dump their DNA into the cytoplasm to replicate. When cGAS finds DNA where it shouldn't be, it assumes a virus is present and activates a protein called STING, or stimulator of interferon genes. STING launches a massive scorched earth immune response. It triggers the release of type 1 interferons and inflammatory cytokines. In the context of a viral infection, this is good because it kills infected cells. In the context of a heart attack, it is catastrophic. The immune system attacks the heart's own dying tissue as if it were a viral reservoir, causing massive collateral damage and driving the fibroblasts to create a thick, permanent scar. However, nature has a built-in brake for this system. It is an enzyme called TREX1, or 3' repair exonuclease 1. TREX1 is a cleanup crew. It patrols the cytoplasm and eats up stray bits of DNA, preventing cGAS from triggering a false alarm. However, during the massive trauma of a heart attack, the amount of leaked DNA overwhelms the natural levels of TREX1. The brake fails, and inflammation runs wild. This is where TY1 works its magic. When injected into the bloodstream or the heart muscle, TY1 is taken up by macrophages, the immune cells leading the cleanup charge. Once inside, TY1 acts as a transcriptional enhancer. It binds to the cellular machinery and dramatically upregulates the production of TREX1. The effect is immediate and profound. The boosted levels of TREX1 rapidly degrade the flood of cytosolic DNA released by the dying heart cells. Because the DNA is gone, cGAS is never activated. STING remains silent. The scorched earth inflammation never happens. Instead of a chaotic war zone, the injury site becomes a controlled cleanup operation. The macrophages shift from an inflammatory state, known as M1, to a reparative state known as M2. They clear the debris without attacking healthy tissue. This allows the heart's surviving cells to reorganize and heal with significantly less scarring. In animal models, a single dose of TY1 delivered after a heart attack reduced the final scar size by nearly half and preserved pumping function. This is a result that stem cells promised but rarely delivered. But the story gets even deeper. The discovery of TY1 also heralded the identification of a new class of biological particle, the exomer. For years, scientists thought exosomes, which are membrane-bound vesicles, were the smallest carriers of intercellular messages. But during the isolation of TY1, researchers used advanced filtration techniques and found particles even smaller, around 50 nanometers wide, that lacked a lipid membrane entirely. These are exomers. They are protein RNA complexes, essentially nature's own nanoparticles. Because they lack a membrane, they are far more stable and easier to manufacture than exosomes. They can travel through the bloodstream, slip through the tight junctions of blood vessel walls, and penetrate deep into dense tissue where larger vesicles might get stuck. TY1 is the first therapeutic exomer. Its discovery suggests that the body has a whole hidden language of non-membranous chemical packets zipping between organs, regulating health and disease. We have only just learned how to read the first word. While the immediate application of TY1 is for myocardial infarction or heart attack, the implications of the TREX1 and cGAS-STING mechanism extend to almost every organ in the body. Consider autoimmune diseases. Many autoimmune diseases are essentially false alarms, where the body thinks it is fighting a virus that is not there. Lupus and Aicardi-Goutieres syndrome are directly linked to defects in clearing cytosolic DNA. In these patients, cGAS is chronically active, leading to constant inflammation. TY1, by boosting TREX1, could theoretically mop up the trigger for these diseases, offering a non-steroidal, non-suppressive treatment that targets the root cause. Then consider ischemic stroke. Like the heart, the brain suffers from ischemia-reperfusion injury. When a stroke occurs, neurons die and spill DNA, triggering neuroinflammation that often causes more damage than the initial clot. TY1 could potentially cross the blood-brain barrier, or be engineered to do so, to limit brain damage after a stroke. We must also look at organ transplantation. Rejection of transplanted organs is often driven by the recipient's immune system reacting to danger signals from the donor organ. Pre-treating an organ with TY1 could silence its inflammatory screaming, potentially improving acceptance rates. And interestingly, viral reservoirs like HIV are also relevant. The HIV virus fears TREX1. The virus needs its DNA to hang around in the cytoplasm long enough to enter the nucleus. If TREX1 levels are too high, the viral DNA gets eaten before it can infect the cell. While TY1 is currently being studied for tissue repair, boosting TREX1 could act as a potent antiviral strategy for retroviruses. We are living in the golden age of RNA. First, mRNA showed us we can use RNA to make proteins, as we saw with the COVID-19 vaccines. Then siRNA showed us we can use RNA to stop proteins. Now TY1 represents a third pillar, transcriptional healing. It uses RNA to modulate cell behavior and enhance the body's intrinsic repair mechanisms. The journey of TY1, from a curiosity in a petri dish to a potential blockbuster drug, is a testament to the power of following the biology. It teaches us that the body often knows how to heal itself. It just needs a nudge. In the case of the ischemic heart, the machinery for repair, which is TREX1, was there all along, overwhelmed by the scale of the injury. TY1 is simply the reinforcement that allows nature to win the battle. As Cedars-Sinai moves TY1 toward clinical trials, the medical world watches with bated breath. If it works in humans as it does in mice and pigs, we may finally close the chapter on the broken heart, turning a fatal scarring event into a manageable, healable injury. The era of patching the heart is ending. The era of healing it has begun. Thank you for listening to G Fun Facts Online.